video was 720p, so it appears very blurry. Even though the last video was me taking, this section in actual real time, since I've already done that, now I will just try my best to explain things. So this one basically, and let's see, it's 8:57 now, so I'm going to try to do this. My goal is 20 minutes, so let's see. So okay, they're basically saying that toxic cyanide, this thing basically binds to Cytochrome C Oxidase and prevents electron transfer. So what would happen to these types of cells if you did the same thing? So, the Cytochrome C Oxidase, it's basically part of the electron transport chain, and it acts as an electron carrier. They all do. Ultimately, it goes to the final acceptor, oxygen. But if you mess up that, right, mess up the cytochrome electron carrier, it's not going to transfer the electrons to oxygen. So, it's going to basically, so if we look at these three things, let's, start with Roman numeral 2, reduced intracellular ATP, yeah, because, you're going to be making the predominant amount of, the majority of the ATP through the ETC, with, the proton pump and everything. So yeah, 2 will happen. 3 will happen too, because without energy, the cell will die. But does it really need more oxygen? No, because it's not like a chemical reaction that, Le Chatelier's principle plays a role in. It's just a broken electron transporter. So it's just not going to reach the oxygen. But the oxygen isn't going to, there isn't going to be, it's not oxygen that you need. You need working cytochrome C oxidase. So that's why it's going to be this C. Here, they measured oxygen consumption and intracellular NADH levels in these types of cells after exposure to this thing. So that's when I highlighted that. It seems to basically mess up cancer cells when there isn't glucose. The mechanisms aren't fully understood. We know for colon cancer, however, it basically activates this protein over here. But we're looking into these types of cells. So let's, so they're trying to see, like, how it works for these types of cells. So first they tested these types of cells with the CK, this protein that works, for colon cancer. That's test 1. The results? Nothing. So basically CK1 alpha doesn't have anything to do with BPC-ML. So forget about that. Experiment 2, or test 2, they needed to localize where this perivinium goes in the cell. It goes to the mitochondria. Okay? And we all know what happens there. MR, mitochondrial respiration, right? Test 3 basically involves how sensitive these cells are when they have no mitochondrial DNA, meaning that they cannot perform mitochondrial respiration. So what happens then? They become, so the cells that don't have mitochondrial DNA are less sensitive to this drug, which makes sense because this drug goes straight to the mitochondria and really, like, in the mitochondrial DNA. So, okay, so basically after they exposed it to this, what happens? So basically, it goes to the mitochondria and it basically breaks down mitochondrial respiration. Like, it prevents that from happening. That's why it's good at killing bad cells, like cancer cells. So, okay, so when you break cellular respiration, essentially, aerobic respiration in particular, what happens? Well, first of all, intracellular after exposures, what do they most likely observe? Oh, okay. So it broke everything down. So O2 consumption isn't going to happen. And NADH works to oxidize things. This is a reduced form of NAD+. And oxidizing things is, along the catabolic pathway, which happens when you're basically involved in, cellular respiration, basically. So you're going to have less O2 being consumed because basically it breaks, that entire thing up. And you're going to have an excess amount of NADH. So, yeah. They elaborate they have the details of how the NADH supplies electrons specifically to the ETC. I mean, that's the ultimate place that it goes. So that's why this is this answer. I'll try to go faster. Compounds that decouple the proton gradient. Alright, so this is basically, things that like make it so the proton pump is no longer coupled to the ATP synthase, which uses that, proton gradient to create ATP. Okay, so what would it have? What type of effect would it have? Dramatically increase, dramatically reduce? Modest increase, which is more closer to A, which, by the way, is never going to be an answer because it's too similar to A. It's not going to have any strong effect at all. Because this drug basically just breaks the entire electron transport chain, so it's not going to even do anything regarding to pumping protons out into the intramembrane space. All right, here observation that it's more effective. This drug is more effective against these cells than colon cancer. If it was true, how would that what would explain that? Colon cancers rely less on. Oh well, okay. I'll just go through these. Colon cancers rely more on mitochondrial respiration for energy production than many other cancers. Well, that may be true, but then many other cancers, that's what makes it not true. We're making a direct comparison of this type of cancer, this type of like leukemia. It's wrong for that reason. What about this one? It's slower growing type. That doesn't really matter. Absorbed into the mitochondria of colon cancer cells. I mean, we don't. Well, it says that. Oh, here it is. This is the answer. It works really well in glucose starvation conditions. So basically, when you don't have glucose, you're not going to be using glycolysis. So there we go. Glycolysis, is the breaking down of glucose for energy and stuff. Alright, this one, hyperventilation just means that you're breathing really fast. So the rate at which you exhale CO2 is faster than the rate at which CO2 is produced in the cells, by the way, by, cellular respiration. So, yeah, that's why it would lead to less blood CO2, right? Because you're getting rid of the CO2 faster than it goes into, your blood. Yeah, this one was a little weird at first. So how a shift in the right results in more oxygen delivered to tissue. At first I was just looking like, for, let's say, like this, right? This is the saturation at this partial pressure. As it moves to the right, it requires more partial pressure of oxygen in order for it to reach the same level of saturation for the hemoglobin. You know what? What the hell? I'm skipping this because I want to finish this fast. Let's see. Yeah, it said somewhere in the passage here about chlorine. Where is it saying that? Oh, here it says. All right, so it says in red blood cell bicarbonate is exchanged for chloride ion. Why do you? Okay, so blood cells in the venous circulation. So if it's in the vein, it's gonna have a lot more CO2, right? And CO2 basically enters the red blood cell to where it becomes carbonic acid, and and then it can leave as bicarbonate, and then chloride goes into it. So, yeah, of course, there will be more chloride ions in the red blood cells of the venous circulation, right? Because there's more CO2 anyway. So yeah, higher concentrations of CO2 produced bicarbonate, which leaves, and then chloride enters. This is... So, okay, I remember how I answered this. So I basically just looked at, like, just what's different and stuff. Because anaerobic respiration leads to lactic acid buildup. That makes things more acidic. Hypoventilation means you're breathing less, so you're not inhaling as much oxygen as you need, and you're not exhaling as much CO2 as you need. So that does the same thing to this. It makes your, blood more acidic. Underproduction of bicarbonate. Remember in the last question, bicarbonate basically, is, like given off from the red blood cell in exchange for chloride, as it processes the CO2, which naturally makes things more acidic. So with less bicarbonate, right, it's not going to go, I don't expect you have to have this memorized, but this is the bicarbonate ion, and if there's too little of this, it's going to go backwards. This is Le Chatelier, and it's basically going to be working, to make it more acidic. So A, B, and C and D are all just would have the same result of making blood more acidic. Carbonic anhydrase, didn't completely remember, but it has to be it because everything else is the same thing. Same thing being different from the carbonic anhydrase, question. This thing over here, whatever. I think I should go over... Alright, I'll try to do this even faster. Oops. Okay, so this they're basically talking about this entire experiment, and they don't talk anything about, it being any experiments being done on, like, like injured nerve cells. And it says in the question stem, this random medical student wants to, apply the results to spinal cord injury in humans. But you can't do that because the study only did it in response to uninjured ones. Yeah. This one, they're talking about... Okay, so MS is when your immune system breaks apart the myelin sheath. So what's the best treatment for that? So, I didn't really want to read everything here. I know that they're making a distinction between CA and AA macrophages. CA seems to be, or induced by exposure to this type of, immune system thing. I think this is like a factor or something like that. While AA ones were through some other thing involved in, the immune system. Let's see, blah, blah, blah. Well, okay, so IL-4 is what creates AA. So, wait, what? Yeah, I don't think it works the other way around. I don't think, like, if IL-4 makes AA exist, I don't think CA, giving this to CA will make it into into AA. I'm actually not sure about that. I don't really want to read this crap. But, okay, myelin sheath. Okay, here we go. Phagocytosis in myelin was greater in CA than AA. And so, we want to stop that. And it seems like it can be stopped with this anti-CR3 antibodies. So we're gonna stop the CA from phagocytosing the myelin by using that right there. I guess I got this one wrong. Whatever. I don't like these types of questions. But CR3, okay, they don't tell you anything. They just say it's an anti-CR3 antibody. So it interacts with antibodies. So it's just going to be, basically an antigen, aka a receptor. This is a kind of long thing. I'll explain that some other time. Oh, this one was tricky, but like it's autosomal, so none of that goes on to any of your offspring. Those are, body, like, chromosomes, not sex chromosomes. Yeah, this one was just basically some Sudoku thing, like, just look for what's, there all the time, or needs, so, like, here we got this crap, TR1, TR3, right? Here we have PG1, we have PG5 again, but we have TR2. So here we have TR2, TR1, no PG5, so PG5 doesn't matter. This just shows PG nothing matters. And since we haven't seen TR7A anywhere up here, basically it's going to be TR2 or TR1. Since TR1 here is with TR3, that's another possibility, so let's see. This PG stuff doesn't matter, TR2, so TR2 is a definite, necessary thing here. Here, remember these things didn't matter? TR3, bam, that's the one that matters. Here, TR2. So yep, those are the ones. Okay, this is another question where you can answer just by looking at how, everything does the same thing except for this. Like, basically, because I kind of forgot exactly what calcitonin did. Like, does it, because because it can do two things basically. It can basically take calcium from your blood and put it into your bones, or it can take calcium from your bones and put it into your blood. So this is going into your bones. This means it's less in your blood. So where is it going to go? It's going to go to your bones. I don't know how metabolic rate has anything to do with it, but this is the only thing that's opposite. This is breaking down the bone. Okay? Osteoblast will do these types of things. And, and this is how a question can really teach you something that you might have forgotten. Like calcitonin, I forgot. Now we know that what it does. It does that. So I think parathyroid hormone is the thing that does the opposite of calcitonin. Okay, this, I just looked at VDRs and, E's, upstream from genes, coding for this crap. So, yeah, so that's basically going to be a promoter. Operator, no. Receptor, no. Promoters, those are just things that are upstream of like the, place, basically, I suppose, where the gene is. Okay, this is where I looked at axon number and myelination, which is basically this G ratio is inversely proportional to myelination, by the way. So here we have axon number. We're comparing it to, the control that hasn't been injured. These two have been injured. Vehicle is just placebo. I don't know why they're calling it vehicle. So vitamin D in injured nerves causes, more axons to develop. So, yeah. And lower G ratio, which means more myelination. So okay, this basically doesn't tell you much about this increasing and both of these things increasing when they are. But it's right because it's a response to injury. That means we're comparing it to the vehicle. Because here, the control did not undergo surgery. And then there's a lesion which removes part of a freaking nerve, right? So to those people, aka the ones that were injured, they were given some placebo crap that they call a vehicle, or vitamin D. So okay, if we're looking at this, MRR and all this stuff, this doesn't, there's nothing really happening here. And then, okay, and this is just, this is an easy one to mess up if you mess up the definition of the G ratio. So this is very easy to pick. And then this one, axon number sharply decreased compared to controls. No, not at all. Axon number is super high compared to, in the control. Oh wait, sorry. Sharply decreased compared to controls in response to vitamin D treatment. Okay, that is true, actually. The tetanus threshold and myelination remained stable. Myelination seems to be kind of stable, but tetanus threshold, that was this over here. Sharply decreased compared to controls in response to vitamin D treatment. But the tetanus threshold seems to be about the same. I think this and this are the only ones that are statistically relevant. This and this as well. I wonder why. B and D incorrectly compare the vitamin D in control groups. Vitamin D in control groups. Axon number sharply decreased compared to controls in response to vitamin D. Okay, you can't do anything with controls here because you can't make any comparison because they weren't injured. I guess you can't compare apples to oranges. I mean, you should know this. How is this tough? What the fuck? This is straight up discrete. Everyone should know this, okay? Unmyelinated axons are 0.5 to 10 meters per second, and myelinated ones are like 150. G ratio to complete lack of myelination. Again, this you would get wrong if you messed up the G ratio thing, because again, it's an inverse kind of proportion. So, basically it's the so okay, if there's no myelination, so G ratio is the ratio of the axon diameter to that of the outer diameter of the myelinated region, which is going to be obviously thicker, right? So it's going to, as myelination goes up, it's going to get smaller. If there's no myelination, then it's going to be this outer diameter of the myelinated region. There's no myelinated region. It's going to be the same thing as the diameter of the axon, hence 1. Okay, so this, straight up, like Wolbachia or whatever, this is a bacteria, and I answered it because I figured vaccination only works for, viruses or something. Endosymbiotic. Oh wait, no, JK. It affects the germ cells of nematodes, and I guess we get it. I don't even know. Least lymphatic fili. Okay. This, okay, lives in nematodes. Evade detection by the immune system. Only when the nematodes are killed does the immune system recognize Wolbachia is there. Okay, so okay, it infects the germ cells of the nematodes, and then somehow we get the nematode eggs in us, I suppose, and then we get fucked. So if you're vaccinated for the bacteria, it won't do anything because our bodies are really just, it's the nematodes that cause this disease over here. So, yeah, that's not going to really work. I guess on that one I didn't have time. This one, well, I mean, yeah, doxycycline works, on bacteria, and bacteria have different types of ribosomes. And it works by affecting the A site of, this type, that subunit. Okay, this one. Jeez, alright. This one is pretty obvious. This one, looking at kind of the opposite things. This is like, just one short muscle spasm and then nothing. Inability for it to contract is just straight up nothing. Transient weakness, compensated, that's like nothing for a little bit, but it's back to normal. This means it's just constant contraction, and calcium channels open up and basically start the process of releasing the acetylcholine into the synaptic cleft. And if calcium is always there, the acetylcholine will keep going, and it'll be this type of thing. Same kind of effect for like sarin gas and nerve gas, acetylcholinesterase inhibitors, except this is more of a calcium reuptake inhibitor kind of thing. Yeah, this one, I mean, I'm straight up size has really nothing to do with it. I'm just looking for polar, residues. And, I kind of forgot what these meant, but I knew that this was valine and this was phenylalanine, and those are nonpolar, so I was like, yep, it's going to be these guys. But no, they're pretty straightforward, actually. Sarin, threonine, glutamine, I think. Yeah. Oh, yeah. Yeah, I didn't remember those. Yep, this one you should know. This is out. Why are these tough? These are not the tough ones. But anyway. Yeah, dude. So testosterone basically goes to the circulatory system. It's lipophilic or hydrophobic, and our blood is mostly water, so it needs to be bound to plasma protein, probably albumin or something. And yeah, it's not going to stay in the nucleus where it was made, and it's not going to, if it was like a non-polar, if it was a polar one, like a peptide hormone, yeah, it would do the second messenger thing. But no, it goes straight into the nucleus. Okay, I don't want to do that. Well, fluid-filled cavity is going to be a blastocele. Gastrula, I think, is hollow. No, that's when the 3 layers come in. Okay. Anyway, whatever. Blastocele. Okay. Maybe you can remember that, but whatever. Oh yeah. Okay, this is a lot to kind of explain. So I'm gonna skip this for now. Okay, if it migrated closer to the cathode, which is positive, it's going to be something that's negative. So we're going to look for... Oh, damn it, I forgot. All this gel crap uses electrolytic cells, so shit's the opposite way. So it's the one that's positive. I just look for what's different. I know this is glutamic acid. I know this is aspartic acid. Both of these are going to be negative, so can't be that. This is not like neutral, so this had to be it. It's just different because it's positive. I don't want to do this. Oh, this one is, I just looked at this, and then it's the same kind of thing that we talked about earlier. Cytochrome C Oxidase gets effed up, So aerobic respiration is not going to happen. So what's going to happen? You're going to get muscle aches through the lactic acid buildup, which happens when, because it's anaerobic respiration. You're going to be forced to do that if you stop letting aerobic respiration work. None of this other stuff makes sense. This was a weird thing. I read it fast, but it's pretty interesting. Apparently, some mitochondrial DNA or mitochondria from the sperm actually get into the egg, but it gets marked by ubiquitin to get destroyed. So, there we go. Well, I know 2-chloropropane means that, it's the 2 means that it's not, at the end of anything. So it's secondary, and you should also know, that alkanes, you should know, how when things get more oxidized, basically, alkanes turn into alcohols, which turn into, aldehydes or ketones, which turn into carboxylic acids, and then eventually turn into CO2. Okay. Yeah, this is pretty easy too. Basically, this dude, whatever, is trying to make amino acids and sugars. Amino acids are going to be L, sugar is going to be D. But he gets racemic mixtures, which means that he has L and D of, both of these guys. But the body only makes single and enantiomeric form, and that's because enzymes in the body are stereospecific. This thing sympathomimetic. The sympatho part, sympathetic nervous system. Mimetic means that it would probably work to mime it or to be similar to it. And it looks like a catecholamine, which, involves dopamine or epinephrine, whatever. This is a... Anyway. You know what? I didn't even look at that when I answered this. All I looked at was constriction of the pupils happens with a parasympathetic nervous system is activated. Increased gut activity, parasympathetic nervous system. Increased salivation, parasympathetic. Increased heart rate, sympathetic. Just, this is like the third or fourth question that you can answer simply based on what is different. So some dude's plate is making some bacteria, and some area that's something that's been contaminated is clear, so the bacteria is gone. So it's a phage in the lytic phase, and you should know lysogenic and lytic cycle. You need to know that stuff. Okay, this one, I just looked straight down into the thing, the FAM83H, and it says single nucleotide. So this thing over here, FAM83H, mutation causes change in a single nucleotide. It's important to know more than that, because if it's a single nucleotide that doesn't change the amino acid, it's like a silent mutation, but it replaces the amino acid from serine to threonine. That's called a missense. Frame shift is if you have, addition or deletion not in threes, because the codon is three nucleotides, so it messes up the reading frame. Nonsense is when it makes a stop codon, so it just stops the protein from being built. Deletion is gonna mess up the mess up the reading frame as well. But anyway, it's a replacement kind of thing. Okay, so it's an untranslated region. That's what that stands for. I'm sure it says it, but you should definitely know it. Yeah, I don't know where it says it, but, well, I mean, non-coding regions, but anyway, UTR means untranslated region, okay? So, it's transcribed, it's not going to be translated, though. So, this is saying it's always both, this is saying neither, and this is... So, okay, this is kind of the opposite. Pre-mRNA post-transcriptional modification makes the 5-end UTR added to it, in addition to a bunch of like adenines on the 3-end. Poly-A tail, I think it's called. This crap, forget that. Yeah, I got this one wrong, because whatever. This one, yeah, I did fast, so my math wasn't perfect. I was basically just doing like coding region plus coding region, gives something like 600, coding region plus coding region, something like another 300, like 900, and then this, like 1200, so I just chose that. Oh, okay, I guess I didn't include the 5 UTR and the 3 UTR. Apparently how many base pairs will be in mature mRNA? Ooh, because mature, so from pre-mRNA to mature, they get rid of the introns, right? But they actually add the whatever UTRs. So yeah, I guess that's why I forgot to add those, whatever. This one, okay, this one is actually kind of tough. I don't know why it's medium. It's definitely, much more tougher. Because the thing is that, okay, so the math part at first is kind of easy because, first of all, you should know histones and DNA and stuff. DNA wraps around a histone. They don't really have a good picture of it. I don't know if they do here. They don't. Wraps around a histone, right? And 200 base pairs of it, right? And whatever. If a nucleotide is 0.3 nanometers, you just multiply this by that. Like, just think of it as 3 times 2, which is 6. And then, add, the decimal places. So you get 60 nanometers, essentially. And, Oh, yeah. The weird thing about it, though, is that when the DNA is wrapped around a here, when it's wrapped around, so this is DNA wrapped around the histone, right? 200 base pairs, which is 60 nanometers. This is the diameter of the histone, 11 nanometers. The diameter of the DNA is 2 nanometers. The thing is, the DNA wrapped around the histone means that the DNA is on the front and the back. That's actually tricky. So it's double the thickness. So 2 nanometers times 2 is 4. And, if it's hard to see here, I made like a bird's eye view. So, this is the histone yet again. And this is, DNA wrapped around. So 11 nanometers diameter of the histone plus 2 for the diameter of the DNA. But it's wrapped on the front and the back, so that's another 2. Total 15 nanometers. So they're asking percentage of the original length is it shortened by. So original length 60 nanometers condenses to 15 nanometers, which means it's shortened by 45 nanometers. And so, they're asking again, basically the ratio. Percentage is just ratio times 100. So ratio of the original, of how much it's shortened by the original. So it's shortened by 45. Original is 60. That's just 3/4, 75%. So tricky. Okay, this one, total length of DNA. So we know here, where is it? 6 DNA, 6 billion nucleotides. All right, we know here a nucleotide is 0.3 nanometers. So back to here, total DNA size, 6 billion nucleotides. One nucleotide, 0.3, nanometers, which is 0.3 times 10 to the negative 9 meters. Just multiply that crap, 1.8 meters. There you go. Bam. Here, I just literally looked at, like 18 is the only one that has much less genes than base pairs, so I just chose that. This over here, so okay, definitely need the DNA helix to be the least compact, so in the beginning, so B and C are out. Heterochromatin and euchromatin, euchromatin is more loose, but I don't expect everyone to completely get that, because usually you would see things talking about just chromatin. But you need to know about the nucleosome. Nucleosome is basically, like multiple nucleosomes create the chromatin. All right? So nucleosome is the most compact because it's literally just smaller. It's not even a fair comparison. So that's going to be at the end. So, as long as you get the beginning and the end, you get the answer. Oh yeah, this one. So, okay. So at first I was thinking Titan because I know that is in cartilage. No, wait. You know what? It's what lets things stretch. And I think the official name for it is the longest word in the world. Okay, it's in the muscle. Whatever. Anyway, let's see. Structural integrity. Why did I choose? Oh, okay. I know that myosin is made of microtubules. I'm pretty sure. Yeah, myosin, right? They're made of actin and myosin. Anyway, whatever. I'm pretty sure I'm right here. Yeah, because it's like the thick. Anyway, so I just chose intermediate filaments just from like guessing and process elimination. This one, you should know this. You should know how reflex arc works. This one, I just didn't want to do the math. First of all, I know that basically color blindness is something that women basically never get. So first of all, it's not going to be A or D. And then I was just between 4% and 8%. I knew, this is Hardy Weinberg and stuff. but I think there's something like, there's like, p squared plus 2pq plus q squared, and I know 8 squared, or like rather whatever, 0.08 or so, I don't know, is like 64. I don't know. I just, whatever. I chose wrong, I suppose. Oh, and that's it. So